Hello, this is Professor White, and today I'll be discussing about the DNA and DNA replication. But before we start, I just wanted to ask you this question. Why do we age? So this is kind of like a phenomenon that we're going to revisit after we talk about the DNA. And aging is actually the same as senescence, or the condition or a process wherein we age. And of course, this has to do with the loss of the cell's power of division and growth. And the answer might lie in our DNA. But we're going to discuss more about the DNA and how it was established as a molecule of heredity. Now, recall in Mendelian genetics, they knew that traits can be passed on from parents to offspring, but whatever that is, they don't know at the time. So, because of this, they need to know more. So it needed to be like, okay, what it is. So what can you give us? So how was DNA established as a molecular particle of heredity? Well, it is able to store information that pertains to the development, structure, and metabolic activities of the cell, as well as stable so that it can be replicated and able to undergo changes or mutations. So some scientists felt that protein was the molecular heredity based on the fact that there were 20 known amino acids. And there are several experiments that have occurred in order for scientists to learn more about the DNA. We will start first with the Swiss physician Johannes Friedrich Meister. He isolated the chemical he called nuclein from the nuclei of a white blood cell. We now call this nucleic acids, which is, of course, your DNA, deoxyribonucleic acid, and your RNA. So he studied and found out these molecules. So these molecules are nucleotides, which contains phosphates and nitrogen base as well as sugar. And the nitrogen bases are your adenine, guanine, cytosine, and thymine. Now, adenine and guanine are purines, and thymine and cytosine are pyrimidine. So they found out, there is a particle inside the nucleus, so let's find out what it is. So because of this, other scientists were curious about what's in there. So Friedrich Griffith experiment basically isolated this virulent strain of a bacteria, wherein he say, okay, well, there's an S strain and the R strain, and then they try to find out whether the mouse will die or live. So the R strain is a non-virulent, so it actually didn't kill the mice. But the smooth strain is a more virulent one, causes the mouse to die. If they heated the smooth strain, which is a virulent strain, it actually didn't kill the mouse. And then if you put together the rough strain, the heat killed one, and the smooth strain, the mouse died. So which tells scientists that there must be something going on in terms of like what caused this change in the characteristics that's kind of like passed on in this particular bacterial strain. So they knew more about a trait or an information that passed on, causing it to be virulent or not. So the follow-up study about the bacteria is your bacteriophage, which is studied by Hershey and Chase. So they experiment on bacteriophages, finally have shown that the molecules of heredity was the DNA and not protein. So that is basically the reason why they have found out about the DNA having that information passed on. Okay, so the conclusion is that the radioactive tag of the DNA went into the bacteria and not the tag protein. So this is actually their proof that there is this DNA in the nucleus. And of course, they say, okay, now we know DNA is there in the nucleus, so how does it look like? So these are the scientists that found out, and like, okay, let's find out what it is. So Rosalind Franklin, Maurice Wilkins, Erwin Shergaff, James Watson, and Francis Crick were the ones that studied and really looked for evidence of how DNA looks like. And this is actually one of the most famous images of the DNA, which is why we know that the DNA is a double helix. So that is based from their findings. So there's a lot of experiment that has proven about what this DNA is found in the nucleus, which is very interesting. So the DNA, going back to here, is double helix, wherein there's each step of the ladder where your nitrogenous bases are found. A partners with T, G partners with C. So that's actually Shergaff's rule of base pairing. Now the sides of the ladder are composed of sugar and phosphate. Now both sides are held together by nitrogen bases. And each side is actually anti-parallel. So when you say anti-parallel, it's kind of like other one is side up and other one is upside down. So the numbers represent the carbons attached in a ring to make deoxyribose, as you can see on the pattern here. So the 5 prime, 3 prime ends basically just an opposite strands, and the line in between the G and the C here in this example basically is that your hydrogen bond that bonds them. Okay, so a nucleotide is composed of one base, which is your base pair, your deoxyribose, which is the sugar, and one phosphate. So that is one nucleotide. Okay, and also to add to some animation over here, as you can see here, that is how antiparallel looks like. See, 5 prime, 3 prime, 3 prime, 5 prime. So let's now move on to DNA replication. So one of the criteria for the molecule of heredity was it needed to be able to replicate or copy itself and pass that information onto new cells created. So this process is not perfect and can result in errors in the new copy, and these errors are called mutations. Now, some mutations can result in an increased number of copies of some codons. Codons are like 3 bases at a time. These are called trinucleotide repeat expansions and result in repeated regions. So as you can see here, we'll explain that in a bit. So DNA replication is the process by which the DNA makes copies of itself and occurs during the interphase prior to cell division in eukaryotes. Now, replication is called semi-conservative because 1/2 of the original strand is saved or conserved. So this reduces the chances of errors. So let's take a look at what semi-conservative replication means. So if you have this original strand, the blue one, then it replicates the blue and the green, which is basically one is old, one is new. That is your semi-conservative. And if you can take a look at this one right here, so this is an animation of how there is a splitting of your DNA, and as it splits, it creates the new partner or new pair in order for your new DNA strand to be replicated. So now you have 2 DNA strands. So let's just break it down with that process that you see in that little animation. So the DNA helicase is your replication fork. So it's kind of like cutting. Now the DNA polymerase, once it starts this replication fork, adds nucleotides and binds the sugar and phosphates. Now DNA polymerase travels in a 3 prime 5 prime end, which is known as the template strand. Now one side is the leading strand and it follows the helicase as it unwinds. The leading strand, no problem. It adds all those nucleotides as this polymerase travels there. Now the other side is your lagging strand, and it is moving away from the helicase because it's in the 5' 3' direction. So there is this Okazaki fragments which are bound by DNA ligase, wherein it patches nucleotides in there. But the problem is that it reaches the replication fork, but the helicase is moving in the opposite direction. It stops and another polymerase binds farther down the chain. So what will happen to the lagging strand? Let's take a good look here. So in this case, your leading strand down here below, the 3', 5', there's no problem. The DNA polymerase will start filling in those nucleotides and so on, going to the right. But the top part, which is your lagging strand, this is where your Okazaki fragment try to fill in part by part on that other strand. And then the ligase is the one that kind of like seals them as they fill in those nucleotides. So that's basically what is happening. So I want to show you this video real quick, just so you guys can see how the leading and the lagging strand gets their nucleotide. So what could actually happen when DNA replication is happening? Well, DNA can undergo some changes or mutations. Now, sometimes when DNA replication is happening, there's actually some repairs happening in these changes as the DNA is replicating. But if there are different types of mutations. You have your substitution, wherein one base is switched, resulting in a new protein, insertion or deletion, which is kind of like a frame shift, which alters the reading frame or the sequence of your gene. Also duplication, which can amplify the gene product, and CNB or copy number variation. It's kind of like a trinucleotide repeat expansions. And one example of that is your Huntington's disease, which involves a DNA segment known as CAG trinucleotide repeat. Now people with 36 or 39 nucleotide repeat of CAG result, may or may not develop this disease. However, people with 40 or more repeats are almost always will develop this disorder. So that's just an extra. Now let's go back to our phenomenon earlier at the beginning. Why do we age? So there is this cap at the end of the DNA called telomeres. Now each strand is protected by this plastic tips at the end, kind of like the ends of your shoelace. Now it gets shorter and shorter each time a cell copies itself. And eventually, when they get too short to do their job, causing our cells to age and stop functioning properly. And telomeres actually act as an aging clock in every single cell. And as a result, DNA becomes damaged over time, resulting in loss of cell function and senescence. Now, this effect can also occur in the mitochondria. So just a little recap on some words that you might need to focus on would be the helicase, DNA polymerase, ligase, primase, replication fork, leading strand, lagging strand, Okazaki fragments, and the single strand binding protein, which are the words we used in DNA replication. I hope this helps in understanding the basic information about the DNA and the DNA replication. And I'll see you in class. Bye.